So yes, good morning everyone. I hope everyone is doing fine, staying safe and healthy during this difficult time of pandemic. So at Bionavit, we at Bionavit and Bionavit team always try to, whatever the scenario is, always try their best to share knowledge through whatever platform it's possible, and keeping the same spirit alive. Today we have with us Dr. Zia Chaudhary, who is a clinician by profession, working for as a director professor at Lady Harding Medical College, Delhi. She is also associated with several big hospitals in Delhi, like Dr. RML hospitals, and she has she is also fellow to lot of international association and has been awarded with the prestigious award Prem Prakash Award of All India Ophthalmological Society for the work in the field of stress business. So his her major area of interest is pediatric ophthalmology, ophthalmic genetics, stress business. She has also done his doctorate from University of Delhi in the field in the same field. Uh, she has several books and publications to added to her list, and if I may read her CV, the list is will go on and on and on. So without any further delay, it's my immense pleasure to invite Dr. Zia to open this talk. And yeah, also I would just like to mention that whatever queries you have during the talk, you can just drop in the chat box so that we can have a nice interactive session at the end of the talk with Dr. Zia. So Dr. Zia, over to you.
Thank you so much, Raleigh, for your kind introduction, and thank you so much for inviting me here. It's an opportunity that I really appreciate because this is one of my passions, to work on, my doctoral work that I did on genetics and take it further. So the topic today, as you know, is big data to small information, how to interpret next generation sequencing data with reference to the clinical milieu. As Raleigh said, I'm a clinician and I'm a scientist, so I am uniquely placed to put both things together, both the bench and the bedside. And it is quite an interesting thing to do, actually. I have a heterogeneous audience today, so I have deliberately kept my talk simple. And as you know, to make complex things simple is quite a task. So yes, and I hope you appreciate. And if you have any queries, as I said, please put them down into the chat box, as Raleigh said. And with that, I begin my presentation. I have no commercial disclosures or conflict of interest, though I have some grant support for some of the material that I may present in this study from the Department of Biotechnology and the University Grants Commission, Government of India. That was during the University Grants Commission is, of course, to the University of Delhi when I was doing my doctoral work there. I acknowledge Professor B.K. Thelma, my mentor, who has initiated me into the world of genetics, and a lot of other colleagues from the University of Delhi and my own lab here in RML, and who have actually kind of been, sorry, I think you're going faster than, can I take this back somewhere? It's going on at a speed which sorry, I want to go back. Raleigh, how do I kind of do this? Okay, so I'll just, maybe there's something that we just queued up here. I'll just go back here and join it again.
You have put on time mode or something, yeah?
I can unlock the time mode. So how do I do that?
Just unlock it. Just unlock it.
Where would that time mode be? I mean, I didn't know that this slide had a time mode when I made it. So, okay.
You have to go
to my presentation. I'm sorry about this. So, okay. Where would this be? Could I do this? How would I do this? Raleigh? Okay.
No,
So
is it going its own, or I mean, just don't put it on the total slide mode, just put it on presentation mode.
okay.
Because
So
if, yeah,
yeah.
because when
So,
you're putting on slide mode, next slide will come automatically.
so how do I stop that? Because you see, I have some animations to show also. So that's the reason why I would want it to be on the slideshow. I did.
Yeah.
If you can help me, if Kanti can help it. In the meantime, of course,
Yeah.
I could start on
Alvin,
it.
can you look into it? Alvin, do
Okay.
we have you with us? Can you look into it, please?
How do I get out of
Ma'am,
this
I
time?
think the animations are only for few slides.
Is this the animation which has got me into the time mode? I'm so sorry about this, but I didn't know. I have some animations which I thought would look good on the slide presentation mode. That was the reason why I kind of went on to the, you know. So if you want, I can keep it on this mode too. That's fine with me.
No,
Because
if you want to do, ma'am, excuse me, if you want to do it on the presentation mode, on the right side, you will see a presentations symbol. Just
I know that, but how do I get
yeah.
out of this time mode? That's what, because it's going on its own, and
It's a
it's not
record mode.
recording.
Actually, you need to stop recording. You need to stop recording. Actually, who's the administrator? No, the person can go and stop recording.
Okay.
The administrator can go and
Alvin,
or
can
the
we
host
stop
of the call.
recording here?
Ma'am, this is not linked to recording of the meeting. It's fine, ma'am. You can go with the presentation. If it is going to like slides are moving fast, you just come back.
Okay, so can I go to the presentation mode then?
Yes, ma'am. Yes, ma'am.
Yeah, but then will it kind of go automatically on, because I may have to explain some slides and I wouldn't want it to
That's
go on
fine, ma'am.
automatically.
If it is going automatically, just come back, like press the back button. It will go to the previous slide.
See, on the top there is an animation pane. No, you just have to just untick that on the advanced animation right side. Yeah, animation pane
Go
is
to
there.
animation, then I have to do what?
Animation pane you
Animation
see on the right side on the
Yeah, and
no,
then
yeah, click on that. It will
I
go
click
off.
on the close.
Yeah,
Is that all
it
right?
should go off. Yeah, close. Yeah.
So now should I go to the presentation mode?
Yeah, please.
And should it work?
Should work.
Okay. So
It should
let's
hopefully work. Yeah.
hopefully. Okay, so I'm so sorry about this, but so my acknowledgements, as I already mentioned, goes to Professor Thelma, who initiated me into this, and a lot of other colleagues. That includes Raleigh also. So here we start. Is where do we begin? Now, I put in a lot of Alice into this, one of my favorite characters. So when the white rabbit put on his spectacles and asked, "Where shall I begin, please, Your Majesty?" he said, "Begin at the beginning and go on till you come to the end, and then stop." A very philosophical book, which when you read as a child, you probably do not realize. But as you grow older, you realize the philosophy of it and the way. So what is this? We call it the dark matter of the universe, the world, and then subsequently the country, and then a small little state where I live, and then the state, and that's the blue line across as the river. I stay somewhere to the east of that. I go to the middle of the city to work. Now, if I were a human being trying to trace out where I was going, can I go back again, please? I'll just go on to this and say and leave that be. So if I were a human being going back in time, trying to say that where would I find Zia Chaudhary in the whole scheme of things? If I were to start with 99% gray matter, black matter, and then go on to say finding her among 7 billion people on Earth, and then maybe about 1.3 billion in India, and then about 12 million in Delhi, and then in my house. That's quite a task, isn't it? Suppose you didn't know me at all. That's quite a task. So that's the same when you're localizing or looking for novel genes. 99% gray matter, dark matter, what you don't know anything about. And when you start looking for one particular gene that might be causing something, it's like actually looking for a needle in a haystack. So the strategy, now this is a big management point, which most management people would appreciate. It's taught to them. That is when Alice say that, "Would you please tell me which way are to go from here?" So the cat says that depends a good deal on where you want to get to. So Alice say that I don't care much where. So the cat said that it doesn't matter which way you go. So long as I get somewhere, Alice added as an explanation. So then the cat said, "You're sure to do that," said the cat, "if you only walk." long enough. That's important. So what's happening? Sorry. So one way to locate is to walk long enough, search and identify each point carefully for structure and functions. Coming back to the Human Genome Project. This was initiated on October 1st, 1990, finished on April 14, 2003. It's an inward voyage of discovery with an open mind to locate one and all. The road was long, the time taken was long, a lot of people walked, or that is worked on it, but a lot of somewheres were reached. And that is exactly what is written on the NCBI site when you say, the Human Genome Project was one of the great feats of exploration in history. Rather than an outward exploration of the planet or the cosmos, the Human Genome Project was an inward voyage of discovery led by an international team of researchers looking to sequence and map all of the genes, together known as the genome, of members of our species, that is the Homo sapiens. Beginning, as I already mentioned the dates before, the HGP gave us ability for the first time to read nature's complete genetic blueprint for building a human being. It's a dynamic process. They did not do everything. A lot gets added up, but a lot was given, and that was the reference genome. But I'll subsequently come back to it. So when I came back to the philosophy, the animations are of course not working, so we'll do without them. Too much of animations always complicate affairs, as I've always seen. When you have a philosophy, so the complete collection of genes called the genome is similar to 99% of dark matter, as the functions are not known. You do not know what they do. They're there, but you do not know what they do. The complete collection of genes which code for proteins, and proteins are what forms the basis of life, that is called the exome. The human exome is just 1% of the human genome. All these genes live in separate spaces called chromosomes. So if I compare a country to chromosomes, there's so many countries in the world. So there are 23 pairs of chromosomes. Now I add the word pairs, we are diploid. So it houses about 19,000 to 20,000 protein coding genes. Now, amongst these chromosomes, you have places called loci. So maybe say in a country called India, New Delhi is a loci. That is a specific fixed position on a chromosome where a particular gene is located. And I or my house may be the gene, right? So if you have to locate me, you have to go all the way to find where I am. So I'll take an example which most of my ophthalmology colleagues would appreciate because people know a lot about PAC6. And I do have ophthalmic colleagues who might be, participating. So that is a chromosome. That's the chromosome 11. So if that were an address, and if I were PAC6, what would my address be? So if I say that I was PAC6 over here, so that's chromosome 11. I'm on the small arm, the French petit. It's called the petit arm, house number 13. So my address is 11P13. Good enough. Most of us have addresses like that. So if I'm a PAC6, what do? Like I travel to work in the center of the city. So I indeed am a protein. So I'm part of the human exome. Okay, so I'm known. I'm not a part of the 99% matter. So I'm a master gene that regulates proteins important for the development of the brain and eyes. So what if I were malfunctioning? What would I be doing? So malfunctioning, you can see there's a color change from green to red. I made it red. So aniridia, that is absence of iris, anterior segment dystonesis, microcornea, foveal hyperplasia. Basically, PAC6 regulates the proper structural formation of the eye and many other functions thereof. So if I were malfunctioning, all or some of them may get affected. Now, what if I were malfunctioning? Well, I have neighbors too. I may be malfunctioning on my own, but right on the same address, pretty much like in a house, you might be having a top floor, a middle floor, and a ground floor. I have two neighbors. My next door neighbor is WT1 or the Wilms tumor 1 gene. And somewhere up on top towards P14 area is the BDNF or brain-derived neurotrophic factor gene. So that would actually mean that if by chance all of us are affected, then we might be affecting different parts of the body. So while PACS might be looking after the brain and the eyes, BDNF is largely associated with intellectual problems, and the BDNF is largely associated with many forms of structural and functional problems of the brain. And of course, Wilms tumor, if the WT1 is affected. So if the neighborhood is, the friendly neighborhood is also affected along with my malfunctioning, then it's possible that a lot of organs in the body may be affected simultaneously. And that is one of the reasons why whenever we see a patient with an iridea, we do get a whole checkup of the body done. Now, if I as PAC6 were always malfunctioning, let's put it like that. So would I always cause these disorders? That's the next question which comes into form. So we have a situation when I mentioned previously that we have 23 pairs of chromosomes. So if there is one chromosome 11 that I showed, then it's not one chromosome 11, it's actually two chromosome 11s, the one from mom and one from dad. Now, when I label them as A and B, I'm not going to say which one would be mom and which one would be dad, because it doesn't matter. And I don't want the audience to go with the impression that something is gender specific. It is not. What I just want to emphasize is that one is from mom and one is from dad. So we have two pairs. So whenever, that's a difference between just a terminology, that whenever we talk about a gene from the mom and gene from the dad, no, I'm not making it as A and B, say gene A and gene B, one would be called a maternal allele and the other would be called a paternal allele. So it means the same thing, but when we talk about alleles, we basically mean both the chromosomes, right? When we talk about gene, we by and large mean one chromosome, but they mean the same thing more or less, as you can see. So if I use sometimes a gene instead of allele or allele instead of gene, it means more or less the same thing. But when I'm talking about allele, I specifically mean when I'm talking about both the chromosomes. So why is this important? So we have situation one when you have both of them functioning normally. So that's fine. Everybody's happy. So that's how it is. I'm not malfunctioning at this point of time. But now there is this concept of what is called dominance. Suppose I talk about B. I am malfunctioning, but my partner allele A is normal and is hugely dominant. So if it's hugely dominant, then that my partner covers up for me. So my malfunctioning is taken care of and there is no disease or phenotype manifestation. So that's situation two. This is what happens in an autosomal recessive inheritance pattern, that I may be malfunctioning, but the disease may not manifest, right? Because it is being covered up. But that does not mean that the malfunction does not exist, because it may come up, it may manifest in certain other situations. I would be talking about it subsequently in my presentation. But at this point of time, it is getting covered up by the dominant normal partner, and therefore the disease is not manifesting. So if you see me, you may not see the phenotype or the disease. A disease is, of course, a further extension of the phenotype when we are talking about health and medicine. Otherwise, a phenotype could well be something like a long nose, which may not actually be called a disease. So might be using these terms interchangeably in the presentation. So now, again with the same example, suppose now if I, as the malfunctioning gene, was dominant over my partner now, the opposite situation, that is the normal gene. That means it doesn't matter whether my partner is normal or not. The partner is not able to, mask my malfunction. So the disease manifests. Whatever I code for is not, that work doesn't get done and the disease manifests. So though it is heterozygous, which means that it's not that both of us have the problem, it's only me who has the problem, but me being dominant, now I go ahead and the disease or the phenotype shows. Now the next situation, okay. Fine, I mean, there is some amount of space given to the non-dominant partner. Let's put it this way, that while I'm dominant, I'm not completely dominant. It's not as if I'm 100% dominant. I might be, my dominance might extend from say 0 to 95 or 99%. I'm not 100% dominant. So that's called incomplete penetrance, which means that while, as I already said, while I'm being dominant, I'm not being completely dominant. So the phenotype that may express may be variable. So again, I want to clarify two genetic terms, two conventional genetic terms, that incomplete penetrance deals with the genotype, while variable expressivity deals with the phenotype. They may be a correlate of each other. So if there is incomplete penetrance, it is quite possible that if the incomplete penetrance is, or the penetrance is something like 5 to 10%, I might be heterozygous, but the disease may not show, or the phenotype may not show. So I might be phenotypically absolutely normal. So this may actually simulate an autosomal recessive inheritance pattern. It's only when you do the genotyping would you realize that the person was heterozygous. So it's called autosomal dominant with incomplete penetrance inheritance pattern. Now, we go on to the next situation. It's a concept called codominance. Now, this is something like incomplete penetrance, but since you might hear of this term and you might ask this question, I thought it's worthwhile to put in the slide over there. This is a kind of situation where A is equal to B. Let's put 50%, 50%, a lot of space given to both partners, so that the dominant partner is not really dominant. It's co-dominant with the normal. So if I were abnormal, and if the other person is normal, then there is something like a 50% point. Like as I mentioned, it's a type of autosomal dominant with incomplete penetrance, where the penetrance is approximately 50% of the malfunctioning gene. So you'll have some phenotypes that are presented somewhere, which is in between. So it's a term that has been coined somewhere along the way, but it means more or less the same thing. When we talk about variable expressivity and incomplete penetrance, we are actually talking about something that varies from 0 to 99%. This is somewhere in the middle. Now, what is the concept of pseudo dominance? Okay, so I'm malfunctioning, fine. Okay. But my partner, which till now was contributing somewhat, was there. I might have been dominating the partner, but still the partner was there, right? Or it might have been an equivalent partner. It might have been something which was not letting me express myself completely, but the partner was there. Now, what if the normal partner was not there at all? Say, for example, this is something which you see with X-linked, many of you may have heard of it, with X-linked recessive inheritance pattern also, lionization as it was called completely. There is inactivation of the normal partner, happens in autosomal genes also. So you will have activation of the normal partner, which means that now it's only B which is expressing. So while I am recessive, but since my partner is no longer masking me, I'm getting manifest, and I'm getting manifest as a dominant gene. The pattern becomes dominant because I would be manifesting myself so long as my partner remains sleeping. I would be manifesting in each generation, like I would be there. So that is how it is. So that is called pseudo dominance, because it kind of simulates an autosomal dominant pattern, but being recessive. And this is, of course, the classical autosomal recessive pattern, which requires that both the partner genes have to be affected equally, or both the alleles have to be affected. Now, as you can imagine, that if you have two unrelated people, it is highly unlikely that two unrelated people across the world would have a similar kind of, affected PAX6 area. So usually these kind of this disorder arises from a common gene pool of mom and dad. I'll be coming to this as I show some representative pedigree charts. But this is a very important concept for genetic counseling, because you can actually make a difference over here in preventive genetics. So message number 1 is, as a synopsis, that autosomal dominant inheritance pattern diseases manifest in heterozygous state. It does not require homozygosity to manifest. So if I am the bad gene and I am dominant, then I will show. I may show a little. I may not show completely if it's incomplete penetrance, but I will show the features that of the disease or the phenotype. The clinical correlate of this means that if a disease manifests or the phenotype is present in a heterozygous state, then the inheritance pattern is likely autosomal dominant. This is one of the assumptions that you can make. If the phenotype is absent, however, it does not necessarily mean that the genotype is normal. Bad genes are often beneficially masked by good genes. I already showed you that, you know, I may be bad, but I may be masked by a beneficial partner, and I may not show my features, the features of malfunctioning. So that is far more common in the population than the actual expression of disease. So this is an important concept when we go in further into molecular genetics. And again, to reiterate that autosomal dominant inheritance patterns may demonstrate incomplete penetrance, which relates to the genotype, and variable expressivity, which relates to the phenotype, which may be a result of this particular genotypic change. So 2 questions that you may ask at this point of time is that what determines which allele is dominant, or what determines the pattern of inheritance? The other one is, why do some dominant alleles interact with lesser dominance, resulting in incomplete penetrance and variable expressivity, codominance, masking, etc., etc. Well, at this point of time, I would like to say that it digresses away from the theme of the talk, and that could be a theme for some other talk, some other day, because by itself, it is a presentation. Why do these things happen, right? So I'll not be delving into this further, but I would like to stress that just because we look phenotypically normal, it does not mean that we are 100% clones of the reference human genome without any variation. Now, this is one statement. I'm making this right now. I'll get back to it sometime later in about 10 minutes or so as the lecture goes on. So here is this question. As I said, we can always ask questions. The Mad Hatter asked, why is a raven like a writing desk? And further asked Alice that, have you guessed the riddle yet? So Alice said, no, I give it up. What's the answer? So the Hatter says, I haven't the slightest idea. And how wonderful it is to be able to say that all the time, isn't it? So the message too is that pedigree charting is based on the physical evaluation of a distinctive phenotypic feature, disease present in one family. A bloodline family implies a shared genetic pool. So what we are doing is we are evaluating the phenotype, and this is as old as the time of Hippocrates, where you would basically be saying that the father and the son, or the mother and the son, or whatever, you know, the charting of pedigrees as to families having similar features was done. So this is an ancient tool. It has a lot of relevance today. The common genetic pool of phenotypically affected and unaffected family members is called an informative family. That is, you have a shared, but some of them are manifesting a particular disease and some of them are not. So that is called an informative family. Genotype analysis of an informative family is one of the best filters to delineate putatively damaging or causal genes for the phenotype being studied. Now I'll run you through a few phenotypes. I can hear some noises. Maybe somebody should mute up. I don't know, like, you know. So there is, I'll run you through some pedigree charts based on whatever I talked about by being Pack 6. Now, when you look at this, we will put A as the good allele, big A, and small a as the mutant allele. Now, you can see this particular pattern classical. When we analyze 3 generations, what we are getting is a vertical transmission of that particular phenotype. And as you can see that if you have AA heterozygous, A is the good allele, small a is the mutant allele, and I'm the mutant allele, I'm the bad guy, but still the phenotype is showing. So it is autosomal dominant. Right? So this is clear to one and all. I could be normal and I show this AA, and whenever I show this small a and A, I'm dominant because I'm mutant, or I am the variant which is damaging, and I'm showing the phenotype, right? So now what about incomplete penetrance? Now this is where the mystery lies. I wish I could have shown this on a slide presentation, but I will not take a chance on this because I need to explain this. So please follow me as I show it to you over here. So if you have this particular presentation where I'm AA and I'm showing the disease, right? And again, I'm AA here. You do the genotype on me and I'm showing the disease. Fine. But I'm normal here. I'm still have AA, that is big A and small a. I'm heterozygous, right? And again, if you see over here question mark, you see that I'm heterozygous. You see that I missed a generation. If you look at a pedigree like this, the first thing you're going to be telling is it's autosomal recessive. So That is why we use words called presumptive, likely nowadays, rather than say, yes, it is autosomal recessive, because the more we know, the more we realize that the less we know. So when it was conventionally done, all pedigrees, once again, I repeat, are basically based on the phenotype, but the phenotype and the genotype may not always match in terms of in pure tones. Okay, so as you can see that I'm phenotypically normal, but my genotype is heterozygous, which means that I'm actually carrying a mutant gene. As I said, bad guys can often be masked by the good guys or a good partner. So that is what is happening. I'm not seeing, the manifestation of the disease here, but I do have that particular, that particular bad gene, right? So, and therefore, though it will not manifest anywhere, it's getting transmitted. Same, it will skip generations, pretty much like an autosomal recessive pattern. So this, when you do the genotyping and you explore, you may be able to uncover this. In fact, most of these patterns have been uncovered only after one did genotyping and thought about how did it all occur, that one came to all these variants. Now, this is presumptive autosomal recessive inheritance. You just have one particular person being affected, and you can see that that person is homozygous, right? So it's homozygous. And as you can see that wherever you have, something like this, it forms a kind of a carrier, like you may be having the bad guy masked by the good guy, the good partner. But the point is that you are not manifesting the disease because the good partner is able to mask out the deficiency of the bad partner or the bad allele. So it's only when both bad partners are together or bad genes are together or both the alleles are affected, let's put it in more genetic terms, that the disease manifests. This, as you can see, is rare in a normal population and can well manifest. So if you see somebody having a kind of a sporadic, sporadic person, a sporadic patient having a particular disease, and you do it and you find, a homogeneous mutation over there, you should go ahead and probably sequence the whole family and you'd uncover many facts. And people, though it may not be cost effective, it's only if you do that, that's how it gives you a pointer as to what you should be doing. Therefore, pedigree analysis is a very important tool in the genetic armamentarium. Now, this is what I meant by pseudo dominance. Okay, so you have this person who is your proband, the primary person you've seen, and you see that the person is homozygous, 2 small a's. And you see a whole lot of them around were affected. And you sequence all of them, and you see that they're also equally affected. They're both small a's. That's not possible. You see, if it were autosomal dominant, only one, a heterogeneous, only one of the bad partners was kind of dominant, it's adequate. So if you require homozygosity, for in a presentation which is not skipping, it means that the good partner is completely masked and is not manifesting at all. It's only the recessive partner or the bad partner which is now manifesting and is manifesting the phenotype completely. But since it's not a true dominant disorder, it's called pseudo dominant. And of course, then we have what everybody talks about, the autosomal inheritance pattern, when we talk about a common genetic pool or consanguinity. Now, of course, it might be very small and it might be to decipher, and it might have been better had we been on the slide mode. And I'm sorry for that. But if you see that all the people were affected, see, we look at this, let's look at this family. And in this family, as you can see, that all of them are affected. All 4 are affected. All the 4 children are affected. Right. So if all the 4 children are affected and you go about and do a genotype, you go back to the parents who are also affected, and they are also first cousins. And if you go back further into a history of, say, 6-7 generations, you'll find a lot more people being affected. And that is when you know that this is a common genetic pool. So 2 partners in the same family who could otherwise have hidden the bad allele, if they come together in the child, then it's going to manifest. There is no way out of it. It's going to manifest because now both of them are together. This is classical autosomal recessive inheritance. Why is it important for us to know? Because it is one disorder. You may not be able to stop or even identify the bad guys in a heterogeneous population. The good guys basically protect. And so many of us might be having variations, which might actually not be conducive, but they are masked. So we do not manifest disease. But this is something which you can really do something about. Because, for example, if you have this particular lady who's affected, and she has 4 children because of her partner, she has all 4 children are heterozygous. But if one of them marries a normal person from, say, outside the family or outside the family pool, then they'll have children who have, again, who are basically, 2 of them would be normal and 2 of them might be het. Now, if this normal goes ahead and marries another normal, you have all normal children and you've done away with the disease altogether. So this is where genetic counseling plays a big part. This is preventive genetics. It is much, much better than going and having a disease and looking after things which you can't help. So this is where action can be taken. And in the same vein, I would like to talk about X-linked recessive inheritance patterns. That is, when you talk about here, let's say X denotes a normal X chromosome. I mark that out. Now, X is normal. So if you have a normal lady, then you will be having 2 X's, which are normal. And if you have a carrier, then you'll be having 1 X, which is normal, and minus means that it's not normal. Let's put it down as that. So when you have the XY and you do not have a normal X to counteract, the effect of, you have the bad X around there. So then, of course, the disease manifests, and therefore, women in these conditions are protected against having the disease. They're usually carriers, but the sons show the disease, manifest the disease. Women can also show the disease. Again, as I mentioned, the question of inactivation. If the good X is inactivated, then women would also manifest the disease. And when we go on to X-linked dominant inheritance patterns, here, irrespective of whether, no, it's the dominant guy. So if the X is affected, then it's going to show. So when you have it with a Y, which can't counteract it at all, usually men in such a pedigree die. As I would like to mention that about autosomal dominant inheritances also, that if a mutation or manifests itself in heterozygous state, then it is a significant, it's hugely dominant over the other protective partner. So if both were dominant, then you're not likely to, it's not compatible with survival. And that is what happens in these also. It's not compatible with survival. You do not, for any kind of dominant disease, this is the message, 1 is enough. 1 is enough to cause all the havoc that it has to do. It doesn't matter what the other non-dominant partner is doing, right? So if you have this, the men do not usually survive. The women, because of various factors, that's again a course of, these are all the variations that come through, because you still have some mitigating effect of the normal X on the abnormal X. So they may survive, and however, it may not often be compatible with reproduction, but if they do reproduce, then they may have children who may carry on the same, the genetic material onto the next generation. So depending on what. So the message number 3 is, these were classical Mendelian inheritance patterns. There are many more variations to the theme, as I've mentioned, and there are discussions by themselves. Now, while the pedigree charts are determined by the phenotype, the variable genotype that may be seen on analysis makes the usage of words like presumptive, likely, putatively, etc. more true to reality. So that's why when you see a phenotype next time and you say, oh, that's autosomal dominant, it really may not be autosomal dominant. It may just be pseudo dominant. Who knows? So you need to do the genotype before you actually say that as to what that particular family is. So it's good to use the word, just one word, saying presumptive autosomal dominant. So knowing the genotype aids in preventive genetic counseling, especially for AR disorders. This is very important. And remember that autosomal recessive disorders are usually rare unless the gene pool increases the probability of its occurrence. That's usually rare, but you can make it common if there is a lot of sharing of gene pool within the same family, which might have been done earlier for many reasons, social reasons, maybe even preservations of bloodlines, say, for example, for monarchies and things like that, that you must have pure blood and eugenics and other stuff. But when you talk about genetics, it can lead on to an increased expression of 2 bad genes together, which may manifest in disease. And so, on the lighter tone, the Duchess said, I quite agree with you. And the moral of that is be what you seem to be. Or if you'd like it put more simply, never imagine yourself not to be otherwise than what it might appear to others, than what you were or might have been was not otherwise than what you had been would have appeared to them to be otherwise. So Alice says, I think I should remember that better if I had it written down, but I can't follow it as you say it. So the Duchess says, there's nothing to what I could say if I choose, in a please tone. So then Alice says, please don't trouble yourself to say it any longer than that. I hope you're not saying that to me at this point of time. Now, I'm not stopping at present. So now here it is that now we've been talking so much about things getting transmitted. Now, what is this thing that is getting transmitted? What is this genetic material? What is this code? So I refer you back to this 1953 article, an exceptional article by biophysicists, biochemists in the University of Cambridge. We all know about that, Francis Crick and James Watson, and how they basically structured out this double helix structure as a model based on X-ray diffraction and mathematics of the helix form. It's a 2-page paper published in Nature, April 25, 1953. But of course, it revolutionized the whole of human knowledge of genetics as we know today. So you don't have to be, write too much to be effective. And therefore, genetic got partners. The genetics and biochemistry had been going parallel for a very long time. But however, now after this, the partners that genetics got, it was biochemistry, physics, developmental biology, mathematics, all into the field of what is called molecular biology. I wish the animation had showed and I could have shown it better, but it doesn't matter. The message is clear. Here I would like to again specifically talk about one person I admire a lot, and that's Frederick Sanger. He's one of the 2 scientists who have won 2 Nobel Prizes in the same category, chemistry. He wrote generally about 70 papers in a career spanning 40 years. He's also, again, from the University of Cambridge. 1958 was the structure of proteins, especially that of insulin, and 1980, determination of base sequences in nucleic acids. Both of them are phenomenal work. Again, to say that while numbers are fine, but your papers should have weight. If they have weight, then meaningful weight, then it's not just the awards, it changes the way humans look at life. So in 1977, Sanger et al. Introduced what is called the di-deoxy chain termination method for sequencing DNA, which is also called Sanger sequencing. He worked a lot on this since 1965 on small sequences as to how to go about doing it so that you could do more. Now, which permitted long stretches of DNA to be rapidly and accurately sequenced so that you would know. Now you knew that, okay, you had the double helix structure and you had this basis, so you needed to know how it worked. Sanger on his own sequenced the human mitochondrial DNA of 16,569 base pairs. I haven't yet talked about the mitochondrial inheritance patterns, and in this talk, I would not be talking about it because the theme had to be maintained. Also the bacteriophage lambda. And the Human Genome Project is based on Sanger sequencing. Now, when we talk about the Human Genome Project, now how many DNA base pairs are there in the human body? Well, 3.6 into that many, as you can see, and about 30,000 genes. 20 institutions in 7 countries participated, about 10,000 researchers participated in the United States, UK, France, Germany, Spain, Japan, and China. So 97% of the human genome is sequenced till date. 3% still remain unknown of the dark matter. But what it has provided is a reference human genome. And that is the basis of next generation sequencing, a powerful discovery genomics tool today. Now, if you can sequence in 45 days of the test sample what took the HJP 5,000 days. That's 12 and a half years. 12 and a half years? No, yeah, but 12 and a half, 13 years, right? And also form reference databases, which have exponentially increased by this technology so that you can kind of go ahead and, kind of compare more and more of it. The Human Genome Project, keeps on updating. If you know, so you can understand that the exome is a dynamic thing. Today it's 1%, but the idea is to keep on increasing it into the dark matter, pretty much like, going into exploration into space, so that you know a little bit more. The more you know about the gene structure and the functionality, the more you're going to add on to that exome, of known things, of known pathways. So that is how it works out. So imagine there is so much to be known, 99%, enough for many lifetimes for many of us. So, well, so that's what said Alice. In our country, you generally get to somewhere else if you can run very fast for a long time, as we've been doing. So then the Queen said that a slow kind of country. Now here you see it takes all the running you can do to keep in the same place. If you want to get somewhere else, you must run at least twice as fast as that, which is quite true because most of us have to run quite a few times more to get to do what we want to do. So now we come to next generation sequencing and big data generated by that. So now we know that we have a reference genome. All of this collated to the fact that now we have a reference genome, and that is the basis of next generation sequencing. That is, the test genome is compared to the reference genome. Analysis requires a strategy. Well, why does it require a strategy? Because there is a huge amount of data that is generated through NGS pipelines that need to be interpreted appropriately. And that is where we say that big data needs to be collated to small, important, collated information that can be useful to medicine, health, and genetics. Now, the aim is to identify novel putatively causal genes. This slide is a little bit word heavy. I go back to the statement that I made earlier in my slide, that just because we look phenotypically normal does not mean that we are 100% clones of the reference human genome without any variation. I already mentioned that we keep on updating it for normals and abnormal genes. So if we were to perform whole exome sequencing or whole genome sequencing in a random sporadic population, it would generate a huge amount of non-interpretable data. I'm not even talking about the cost that we pick up 10,000 people and start sequencing them. It would really kind of, we'd get a lot of data and we wouldn't know what to do about it. How much was normal, how much was not. So we need filters. So presence of variants in a subject with the phenotype establishes neither pathogenicity nor causality. And that is why nowadays we don't use the word mutation. We only use the word variance. It's when we have proven a variant to be actually damaging do we use the word mutation. Previously, as you know, and when I was a student, they used to say, oh, that's a mutation. No, that's not a mutation. A variant is not a mutation. A variant can be absolutely normal. At best, it can be interpreted as an identified point from where functional work can be initiated. However, if you find out a variant and you think it could be damaging, then you can work on it. And these could, of course, be experimental, that is animal study replication, or you could be having iPSC stem cells, that is induced pluripotent stem cell research, or cell cultures to evaluate gene expression. NGS technology has given rise to what is called a new branch, and that itself is another topic for talk. And that is the topic of, and that is the branch of bioinformatics, where you are basically doing a lot of analysis, a lot of population statistic analysis, pathway analysis, computational interpretations of the big data with reference to relevant filters, so that you can get to some information as to localize what could be your candidate gene for a particular disease. This is very, very important. However, if the aim is to look for presence or absence of variants already identified as being putatively causal, now we are not doing away with what we had already done earlier. If you have cystic fibrosis and we have the cystic fibrosis gene plus and minus, though there's a lot more to cystic fibrosis now that we know the sequence things, but what has already been done, you just put it in a panel. And pretty much like Sanger sequence, you can do targeted resequencing. Maybe it's a little bit more expensive. Do the same thing and you get to a plus minus. And that is what in clinical terms is what we mean by gene panel. So whenever in a particular setup, we say, oh, let's go and do a gene panel done, an ID panel done. This is what you do a plus minus. You really do not know. You don't know 99%. So yes, but if you really suspect something and you think that you might be able to find out something from what already is in that gene panel, yes, it's good. So many clinicians use this nowadays as an armamentarium, and there's nothing wrong with that. But as you know that 99% things you do not know, even out of the exome, 90% things you do not know. So what you do know till now, as you know, after we have sequenced the human genome, in the past 20 years, the increase in knowledge in this field has been exponential because of the enhanced technology that we have. But prior to that, it was definitively going in a particular direction, but it was relatively slower. And so targeted panels usually take into consideration what was already there or what extra has been enhanced in, say, the past 10 or 15 years. And so it's like a book that comes into the market after a lot of research has been done. The book collates a 10-year data, something like that, So you can say yes or no to it. That's about it. Okay. So that's how we go about it. Now, my aim here is not to talk about how you do a genome sequencing workflow. As I said, I would not prefer to sequence, just random sporadic population. Though that is one strategy. As I said, you've got to be very clear about the strategy. I would prefer to take in families with a common gene pool, an informative family, and that is what I have been doing in my work. Now, the rest is, of course, how you prepare out a library with reference to, the human genome. But our work starts from here, after variant calling, how do you identify the variant? Now, that is done by, by industry nowadays, most of it by industry and by academia, as to give us a library, give us good quality control. I cannot emphasize how important quality control here is, because if quality wasn't there, you'd never be able to interpret that the variant that you were seeing was actually the variant that you want. So now, after that, you go ahead and start prioritizing the variants. So if we talk about that, there are certain assumptions and filters that we have to use. So what is allele frequency? This is the concept. We talked about alleles. So this is a concept in population genetics, and which has come out of being able to form many more databases, which has, of course, been because the human genome after 2003, as I said, there's been an exponential leap, and especially from 2012 onwards, the last 10 years has been totally an NGS era. So the number of times the allele of interest is observed in a population divided by the total number of copies of all the alleles at that particular genetic locus in the population. So that is, it's basically a mathematical computation. And why is it important? It's important because minor allele frequency, or MAF, is the frequency at which the second most common allele occurs in a given population. They play a surprising role in heritability, since MAF variants, which occur only once, known as singletons, derive an enormous amount of selection. The basis of this, I would like to emphasize, there have been other techniques. We have not talked about that right now, because again, that was not the theme, linkage or Jivas. Now, here I would like to stress upon the second point, that Jivas basically came up with common variants, like hypertension. You do 20,000 people in a population, you come up with some parents, you kind of see people who have hypertension, who don't have hypertension, and then you kind of statistically analyze and say, oh, these genes could, actually be, putatively causal for hypertension. Okay, so they're common variants. They're present in the normatives also to a significant degree, or people who may not manifest hypertension at one particular point of time. Again, difficult to interpret, because if I may not manifest it today, I might manifest it 10 years later. So that is an important point to think of. So there are, of course, assumptions are there in every type of study. That's why clarity of how you want to do your study is very, very important. So when we talk about discovery genomics with next generation sequencing tools, have made these rare variants detectable. They were not detectable earlier, because when you're doing a statistical evaluation in a population, you'll come across common variants. And while they're significant, what has been seen, that it has been seen that this particular rare variants, rare variants would mean that I, manifesting the phenotype, have this variant, and you, not manifesting the phenotype, don't have that variant. Okay, so that could be putatively causal for that particular disease. So that is why this becomes very, very important to know how frequent that particular variant is in that particular population. And we have so many databases. So for example, what is more common, what is a common variant in, say, a South Asian population could actually be a rare variant in an African population. So it shows demographics, So this is important when that is where the databases help. Another concept, which of course I would like to emphasize upon, is what is a synonymous variant. Now, there is redundancy in the genetic code, but I call it protection. Otherwise a lot more of us would be having diseases. So there is redundancy in the genetic code where amino acids are encoded by more than one codon. Ideally, you would say it should be one particular message from the DNA onto the RNA and the protein forms. No, there are multiple codons that could code for the same protein. So which just means, now what are we, what is life associated with? Life is associated with proteins, you know. So you may have a lot of variants which code for a single protein. So even if there are minor changes or there are variants, okay, they do not actually cause a difference in the final product that is formed. And we are interested in that final product. So the final product is all right, we go on with life and we go on without disease. So actually it's a big protection. But however, we know that it does not help us in identifying a disease, excepting that if we know that there are synonymous variants, we know we have to shun them out of the system when we are analyzing the rare variant. So here are two slides which I had basically, which I've taken borrowed from my PhD presentation slides. Now, if you took out this particular family, you'd look at it and say, oh, it could be, it is autosomal, recessive. When you see these three, and you see that okay, someone else up there has it also. So you say that it could possibly be autosomal dominant also. So when you did the NGS, or when we did the NGS, we found about 44,000 coding variants that came up. So when we removed common variants, that is after applying MAF of 0.01, and we had about 3,000 variants. Then we removed synonymous variants, that is 1,000 more basically were such which could code it for a single protein. So we knew that is not how you could basically, they would really not be effective in disease causation. And then we basically saw, so you can see that, if you see that there are, this is basically was affected, but we had not even sequenced these three, right? This lady was no more. I mean, when you have that marked, pedigree charting is another talk and the interpretations thereof. But, so you have these who were sequenced. Okay, so this was normal, and this lady was normal from this generation, and you had these three who were not there. So you had 3 affected and 3 unaffected. So when we saw homozygous variants in 3 affected, and we saw that there were 9, but they were present in the affected also. When we put in the filter, the pedigree filter for homozygous variants in 3 affected, but absent in 3 unaffected, we found that our derivation was 0. We analyzed this family in another manner, not going into that. Again, it's out of, sync from the theme of this thing, but that is where I was just trying to demonstrate filters as to how they work. And here is this family. You would say that, okay, this is an autosomal dominant family. We basically sequenced these. As you can see that there are 8 members who got sequenced, 5 of them who manifested the phenotype, and 3 of them who did not. Again, there were about 45,000 coding variants that came up after we did the NGS. So after removing common variants, about 3,000 variants were there. After removing synonymous variants, we were able to do it with another 1,000 more variants. So about 2,400 variants were there. Now, out of those 2,400 variants, we applied the familial filter. That is why I say that the familial filter understanding of pedigrees is so important to NGS interpretations. It is so difficult if you just pick up. Otherwise, you would basically be trying to interpret something out of 2,400 families, 2,400 variants, which is a difficult task, no doubt, right? 2,400. So here what you do is, now suppose we put in the filter this way. We saw that if we took in homozygous, again, as I said, that we are not choosing, we put it through both kind of interpretations. We took in both. That is, we put in where the genotypes were, homozygous for the affected and heterozygous for the affected. If it was heterozygous, then it would be autosomal dominant. Homozygous, then obviously it could be one of the variations of autosomal recessive or an incomplete penetrance, as the case may be. So when we saw homozygous variants in 5 affected, there were 9 people who showed those. But when we put in the filter for saying homozygous variants in 5 affected, but absent in 3 unaffected, then none was, we got nothing. Our derivation was null. So we know that this was not an autosomal recessive condition under any circumstances, or pseudo dominant, or anything as you would call. Because just from the pedigree, as I told you, you cannot really say. You can only say it's presumptively autosomal dominant. But when we put in the filter in the het pattern, in the het with the het pedigree, that is heterozygous variants in 5 affected, we found 97 variants. But when we went further and we saw that heterozygous variants in 5 affected, but absent in 3 unaffected, then only 8 were derived. And we also checked our lab database. All of these 8 were not, in this particular genes that we derived. So, of course, I've not mentioned the genes out here, but these were now our candidate genes. So this is how we applied filters. So then after that, after we identify those 8 genes, for example, if I take that example, or if you take other examples, after you've identified your candidate genes, then you see what type of variant are they. Like, you must have read up in biochemistry. I'm talking about my medical colleagues also, you had missense, spice, frameshift mutations, so I'm not using the word mutation. So all of them are basically variants. Now, bioinformatic tools are mainly used to analyze damaging effects of small changes or mainly missense variants. So if you have splices and frameshifts, you usually get no data because the changes are massive. Okay, so normally we are talking about missense variants under most circumstances to be effective in the comparison and the statistical comparison. So that's one filter. Of course, if you don't find anything, then you have to look for others. But this is the way to start because, as I said, that's how you collate out big data into small information. Then, of course, you have a whole lot of, now this has really been growing by the day, a lot of bioinformatic tools by virtue of which you actually sort out as to what is the damaging effect of that particular variant. The oldest shift in polyphen. Shift is sorting, intolerant from tolerant. A small form is shift. And polyphen shift basically just says looks at the functional effect of the change of one particular amino acid in the actual functioning of the particular function of that particular whatever the gene was doing. And out of this, you see, if you take this whole cocktail of things, like many of these pipelines like KGG-SEC or WANOVA basically give you this whole pipeline. So you'll have a lot of data like statistics. That's what I tell my students also, that you take in a lot of statistics. You might have plus minus and everywhere. You may not be able to get much information out of this mocktail, like it's like putting orange and lemon together. It may not taste very well, right? You got to select out what you want for your particular cause. For example, mutation taster, mutation assessor is very useful for cancer genomics, right? So on the whole, I prefer to take shift, polyphen, catred, and say JERP. JERP is one of those things which shows whether the particular mutation is lasted over different, species. That's another thing, because this is another talk by itself, so I'll not go further into this. You have to see what you're looking for, because if you take all of them, it is possible that what is muted in one might be what is damaging in one, maybe inconclusive in another, and maybe not damaging in others. Then you'll again get to no conclusion. So you should know what you're looking for. Each of them assesses something different, and you should be clear about what you want a particular thing for. Though the advantage of having all of this in KGG-SEC or WANOVA is that you get them all together so that you have those to choose. But finally, it's you. It's not the AI. It's you who's choosing as to what you want and what interpretations you want with logic. That is very important. Other filters that you may use is subsequently after that. So say, for example, you use this and you find the damaging ones, the really damaging ones. Then you go ahead. Now, as I say, that biochemistry and genetics, go a long way. They all came up together, and somewhere along they, kind of now have submerged into what is called the field of, molecular biology. So a lot of genes as to which genes are regulating what was not known, but the pathways have been analyzed in great details over the past 100 years. So pathway analysis, interacting partner analysis as to what gene is doing what, and network analysis of genes. Functionality as per known functions of those genes as to where they're acting. They're all updated in the NCBI site, and they keep on adding up every day. Like you do something, you dynamically add to it. Subsequently, functional analysis, actual functional analysis by methods which I've already mentioned, animal study replication or stem cells or gene expression if you want to do it, or otherwise doing the screen of whatever you may have found in a sporadic population. That is going through with a gene panel where you can do it with Sanger sequencing or with targeted resequencing. So all of this would probably give you pointers into what is your candidate gene. Suppose you've delineated 7 or 8. What is your candidate gene of 1 or 2 out of those which you would like to follow up? So let's put it this way, that if I showed those 2 particular, charts which I've shown, so from 50,000 variants, you get to 2 putatively damaging genes, causal variants, and obviously that's a big step, isn't it? I mean, towards getting 1 or 2, which you can look up further in different ways. However, you may not be able to do that for genomic analysis. Now, there is a reason for that. I was talking about exome now. Now, what is the exome? As I've already explained, that it's that part of the genome formed by exons, so the areas which will eventually code for proteins. So we're going more by functions. When we talk about proteomics, we're going by functions. So the exome of the human genome comprises about 180,000 exons, and it's just about 1% of the human genome, as I mentioned, the dark matter. the dark matter and the light matter. So as the exons are protein coding, hence it's extrapolated that mutations therein can cause diseases, Of course, there might be many things in the dark matter which might be causing a lot of things, but we do not know about them. If we do not know about them, then interpreting them becomes very difficult with a reference genome as we know now. I mean, as human knowledge increases, as I've already mentioned in the last time, that, as we try and increase the exomic area, with increase in knowledge, obviously that is going to change. So if you ask me today that what would you like to do if you are given to analyze something to look for putatively novel genes, then I would take up whole exome sequencing as a hypothesis-free, relatively cost-effective modality of delineating novel genes for different conditions, while I would keep whole genome sequencing for those who formulate databases like 10,000 genomes, 100,000 genomes exact. That keeps on increasing because you would need that. You would need to know what exists structurally by name, and later on go on to functions and talk about it, maybe add on to that. So let's put it this way, that genetics is slowly getting incorporated in the wider concept of what is genomics. The main difference between genomics and genetics is that genetics scrutinizes the functioning and composition of the single gene, whereas genomics addresses all genes and their interrelationships in order to identify the combined influence on the growth and development of the organisms. So the whole gamut of things that we talked about, and that's why it's called genomic medicine, and you can apply it tomorrow in health and disease, in what is called personalized or genomic medicines when you're specific to a particular person. So this change, of course, as the caterpillar asked Alice, is who are you? Said the caterpillar. So as this was not an encouraging opening for a conversation, Alice replied rather shyly, I hardly know, sir, just at present. At least I know who I was when I got up this morning, but I think I must have changed several times since then. So thank you very much for a patient hearing, and I'm willing to take any questions. I think it's been 1 hour. Yes, please. Thank you.
Thank you so much, Dr. Zia.
Hello. Yeah. Can you hear me?
Yeah, Dr. Zia, this is Rumi
Yes.
here.
I'm here. Yeah.
So thank you so much for the insightful talk and explaining about the pedigrees and how to correlate those familial pedigrees with NGS data. Believe me, it's quite a task for us, being not from the genetics background, to understand these things. Thank you so much. It was very, very helpful. And if there is any question, people can raise hand. I couldn't able to find the chat bar here.
Okay, I see somebody raising a hand. It's RP. I do not know. So it's Dr. Ranjeev Patel. So can I have your query? Yes, please. Hello. I saw a raised hand. So what do I do? Should I click upon it?
No. I think he left. Okay, no issues. So any other question from the participant? They can ask, they can just unmute and ask.
Zia, actually, thanks for the presentation and putting in efforts to, praises to the entire genome sequencing methods. And we quite know that genome sequencing is the future. And probably the more India needs a well-developed structure and infrastructure to support such kind of a sequencing requirement currently, because as compared to the other parts of the world, India is far much behind in terms of sequencing methods specifically. So as an individual person, and we have been quite interestingly following on sequencing methods and trying to see what innovations and what methods we can adopt, which can help scale up the, for first bring awareness about the sequencing and the importance of the sequencing, which is driven through the government organizations, which will help us to build infrastructure. Second thing is what sort of a support you and your team can give to develop such kind of an infrastructure if required commercially.
Okay. See, I owe a lot of this to my knowledge of how to go about interpreting it, with a basis on genetics, to Dr. Thelma. And I know that in the beginning, when I fell into the trap of doing the same thing, that you see a phenotype and you try and say, let's sequence it and see, and whether you'll get something. And I remember one statement which I've not forgotten. And she said that if you have a lot of money, which the West probably has, you can do that. And you'd probably get somewhere, as I said, as Alice would get somewhere. And you would probably keep it in a database and see. So I see a lot of studies now in the West. I mean that in the West, because I would also again like to kind of counter what you say to a small extent, that the West is still very linkage based. They still love to do linkage because it started off linkage a long time back in GWAS. And in fact, I would say that next generation sequencing, is done a lot more in India and China than it is done in Europe and the United States, though they are catching up. And that is quite a bit, which is one thing which I observed in the past decade, that they were really not into next generation sequencing technologies to that extent, excepting the groups that were doing a lot of, making out the databases. So maybe the databases didn't get made in India, but then there are a few studies, which have been funded by the government of India, where they're trying to make an Indian database. But I'm not coming to that. What I'm trying to say is that because I'm a clinician, and it's easy for me to, and I see the patients. Of course, I have one specific interest, strabismus, which I look for, and we are looking for that in terms of novel genes. And that was a part of my PhD work also. The point is that if you look for a particular phenotype in a family, I found that to be a very, very judicious way of doing things, So when Raleigh and I interacted earlier, and when she told me this, that is the whole thing. When you are able to put that pedigree filter into your analysis, that is when you actually do away with so many things, because you know what you're looking for. That knowledge of genetics is very important. That knowledge of pedigrees is very important, because as I showed, that you can have many variants. And that actually is a big help, and that is what linkage is about. You put linkage and NGS together, your derivation is much more. And that is what actually this is. It's basically, I haven't talked about linkage, I haven't talked about GWAS, and maybe some other time, if Raleigh permits, we could talk about those in a more specific audience who might want to know more about it.
A detailed conversation on that. Yeah.
And that's what I'm trying to say. So this was more general, because it was how to put it into the clinical milieu as to how it would apply what you see in a patient. So I found, if you apply, when you talk about families, you're actually talking about linkages. You're taking out that common gene pool, which is being taken to the next generation. But in some ways, when you have certain families, I haven't shown examples of mine or the work that I have done or the work that I do, where you might be having just 3 people being affected in one family and nobody else having anything. So those are not even good link. They're not informative for linkage. But in sequencing, yes, you go ahead and do direct sequencing. So that is the policy. You have to have a strategy. I mean, without a strategy, NGS interpretation is very difficult. Is that what I found? But that's what I've been doing for the past 8 years now. And yes, and I would say that applies to every disease. And the second thing which I would like to stress, and I know as a clinician, I have an advantage there, especially if it's the phenotype that I look after is the phenotype, that the phenotype that I take to my, the bench is the phenotype I'm also assessing in the clinic. So bad data recording, if the phenotypic data, like in many of the labs, as I've seen, is that you'll have a lot of, good genotyping being done. And when you go back to correlate with your clinical collaborator, you'll see that the phenotype has not been properly recorded. Now that is wastage of data, because if the phenotype, and that's something in which the West is far ahead of us, because I've seen that being done. The phenotype or the documentation is not managed properly. Then how will you correlate? At the end of the day, as I showed with the help of the pedigrees, that if you do not have that correlation, you'll never know what the inheritance pattern was, and you will never be able to apply that filter to NGS. And so you will always remain, hunky-dory about the results. So that is very important, basically assessing the phenotype properly and recording it properly. I mean, you don't do that, interpretations will always be in the gray zone. There is so much of uncertainty in any case, that these are the 2 main things which I think is important to assess. That's what I thought.
Yeah, that's very true. We at Binamit deals with lots and lots of data, but like funneling those data to make a meaningful sense and interpret it, that's a big deal. And yeah, definitely true. I mean,
I agree. I agree, ma'am. I agree, ma'am.
Phenotype.
That's the reason, because actually we have executed major projects of Sonics.
It's phenotype. Phenotype. If the phenotype is not there, then I think there's a major, just getting the genotype. Just having the phenotype without the genotype is not useful enough. Just plain pedigree charting, you would say it's autosomal dominant when probably it's not when you do the genotype. But having the genotype without having a proper phenotype, that also is not, it doesn't lead you anywhere. I mean, so that is it. Both have to be recorded properly. The genotype gets recorded properly when you do this, when you do good quality sequencing and whatever, and then you recheck it also. As I forgot to mention that, of course, that NGS is always, you always apply the good old Sanger method, or maybe do a targeted resequencing to again recheck your variants. If you found something, you would recheck it. You would not like to just say, oh yes, because you do put some to that extent. Yeah, you would always say that doing it experimentally and actually seeing that particular variant being shown experimentally is the most definitive way of doing it. So yes, but then at least you are not sequencing the whole human genome, the test genome again, and going ahead with it for 14 years. You can't afford to do that. So you just sequence that part where you found it and see whether it's there or not. You can resequence it. You do the Sanger sequencing for only that area. So that is routine. But NGS has helped us so much. If we do not funnel that information that we get into a proper collated modality of how to interpret it, then obviously you're stuck. You'll have variants. So the variants, in fact, somebody asked me that why should you have some, earlier we didn't have so many variants, so we didn't know. So now that the argument against that is, you should have choices. I should have 70,000 variants to choose from, and then I should know how to take out 2 from those 70,000. That's the whole idea. That's how I work towards it, and that is where the the filters and the mind works and the logic works. If I didn't have 70,000, if I only had 2, then that is also not a very good idea, because I don't know about the rest. Then that doesn't mean that they don't exist. That is just limitation of your, so it's good to have big data or a big pyramid, but to be able to kind of go to the top and make it a funnel is what you have to work at. And that is what we need to do. And yes, and good record keeping is the, is the basis of that anywhere.
Yeah. So, I think I see another hand raised by Dr. Raj. You have any question? You can unmute.
Hi Zia, it was a great presentation.
Hi.
This is Kumar again.
Yeah. Hi. It's good to see you, Kumar. Long time.
Yeah.
Kumar's my colleague at UCLA. So it must be pretty much in the night. Yeah.
Pretty much.
Thank you
like
for listening. Thank you for keeping awake.
11:46. Yeah, it's pretty much like midnight.
I can understand.
Yeah. So, couple of questions. I don't know. I'm going to bother you a little bit.
No bother me.
I'm not going to bother you much. So, how definitely you can say that a variant that you find is causal for a specific phenotype?
See, Kumar, the point is that you, as I already said, that, the more we get to know, as I said, the more we get to know how much we don't know. So that's why I've been using the word putatively causal, presumptive autism. I already mentioned that in my talk. See, when you know less, you will say autosomal dominant. But when you know a little bit more, you would like to modify to saying presumptive autosomal dominant, because you really do not know. So similarly here, the logic that you're applying has to be, let's say, how should I say, the logic that you're applying has to be logical. That has to be scientific. That it has to be a scientific basis, that I applied this filter, I applied this functionality. It should not be whimsical. It's not because I like the name of a gene, or I thought that
my second question would be
that's what I'm trying to say.
What's your next step? Like, what's your next step? Like, you identified the genes, you identified the variants.
After those,
let me finish.
70,000 variants came out on their own.
No, I got you. I got your total.
See, out of till that 8,
you narrowed down 8. That I totally.
Let's put it this way. That from 70,000 to 8.
It's amazing. I don't have questions on that. What I'm telling is, how do you establish the fact that
I'll come to
these particular variants will cause a specific
I'll come to that.
phenotype.
I'll
Are you
come to
going
that.
to test it? Are you going to make a model? Are you going to test
Yes.
this models, or are you going to introduce these variants into?
That's where we want your help, as you know, for gene expression. Yeah. So you said that earlier that you're going to be a partner. We need your help for this. Kumar is an excellent, he's faculty at UCLA, and he's a molecular biologist. So let's put it this way: that Kumar, now, from 70,000 to 8 is direct sequencing filters. Okay, let's put it this way. I'm just giving an example. Out of this 8, now we are doing some amount of functional analysis based on, so pathway analysis, but these are all based on logic and presumptions. So that 8 to 1 is actually a step where we actually need to do a functional analysis, right? So that is the most definitive way of seeing. There are 2 ways of doing it. One is a functional analysis. The other is doing a huge mutation screen, that is basically taking jivas into consideration. Like, once again, taking jivas, find your mutation and do a gene panel plus minus on a huge number of people. And then you get some kind of a statistical correlation. But one of the biggest faults of jivas is that it establishes genome-wide association studies. As I say, it establishes association. It does not establish causality. Okay, so that is one point in it. But exactly, I know. So, but then, so the only way in which you can establish causality is by doing functional studies. Now, functional studies, number one could be animal replication. We talked about that you basically, try and replicate it in an animal. Number 2 is, of course, stem cell research, just to kind of see whether induced pluripotent stem cells grow in a particular manner with whatever you have derived with that particular gene derivative, or whatever that you would kind of try, to kind of see whether it works out in that particular direction. And the other is gene expression. That, for example, if I am talking about, say, strabismus, I'm talking about neuronal tissues. When I'm talking about concomitant strabismus, I'm talking about neuronal tissues. When I'm talking about muscular tissues. So I like to take those cell lines and see whether it works on them. And if it does, then that's the model that's going to work.
Okay, that's a nice way of that's a nice way of explaining. I mean, I do understand what you're saying.
But you see,
how do you explain in a tissue, in eye especially, and the strabismus is a part of the eye phenotype. So let's say it could be a muscle, it could be any other part of the eye where the cells have a specific organizational phenotypes, like its own functional phenotypes. Like, it could be any function, it could be movement, it could be phototactic, like a light controlling ones, or you can say sensitive ones, or it could be direction, it could be focusing, it could be anything. All these are phenotypes. So you're sequencing using the blood from the patient, right?
We're using whole blood for DNA.
Whole blood, right? I understand. So there is so much of heterogeneity, or the functional heterogeneity in the cells, in the tissues, right? So the question here is, if you do where the phenotype is occurring, like the origin of the phenotype in the tissue, in the eye, in a specific cell population, let's say, not the whole, let's say, let's take a muscle, let's say a superior oblique muscle in the eye. Let's consider that as it's defective, right?
Instead of blood, you're saying. Instead of blood.
Instead of blood, yeah. So how do you assess that whatever you see in the genome that you have sequenced in the blood, with respect to the functional phenotype that's going on in a specific part of the tissue? Because not the whole tissue is affected, only some cells are affected in the tissue. Otherwise, we wouldn't be living here. We wouldn't be living with any kind of cancer, any kind of thing. And inflammation would have killed us. Viral infection would have killed us. Flu would have killed us. We are surviving primarily because only some cells get affected, not the whole body, or not. So that's one question, number one. Second one is it like a single variant that is affecting, the phenotype or multiple variants? That is.
No. I didn't have the time. I prepared for a 1-hour talk, and there are, as I said, these talks can be bifurcated into many more talks, and the oligogenic and the diagenic. So I'll come to that. So first of all, your first question was that why should we not pick up a specific tissue? So specific tissue, I would say at this point of time when we know nothing goes, I would like to pick up a wider base. The reason for that, and that's the reason why blood would probably be better. The reason is because we still do not know whether concomitant strabismus is a function or is a malfunctioning of something present in the muscles, the extraocular muscles, or the brain. So when we do not know, it's better to kind of take something wider, as I say, take a wider perspective, a wider base, and then go on and see whether the particular genes that come out, out of that analysis actually have functions that are related to muscles, or so much that we don't know. I've already mentioned that, but whether what we find out has some relations, and then go ahead and see whether there is expression in. That is the way I would like to do it. I would not like to modify my methodology by picking up a tissue right now, though alternately it can be, alternately meaning simultaneously it can be done. Like, for example, if you decide to do that and we can add on the results and we find that they match, that yes, I would say definitely that's a great kind of an outcome. That is where it works out. If simultaneously you're able to pick up muscle and you're able to do that.
If you take all your 8 genes, right?
Yes.
And its specific proteins.
Yes.
All you need is a section of a whole live human.
Yeah.
And then try to find out where does it express exactly. So
exactly, that's what we do by gene expression studies, isn't it? I mentioned that.
It's as simple as like 1 week.
Already available in NCBI and other such softwares, so we've been doing that.
Okay, cool.
But you see, one is to actually do it physically, the other is that you can find gene expression. That's the other way. That is what we have been doing as a filter, so that you know which one to pick up more, which one shows more expression, which tissue. So if there is something that shows more expression in, say, nerves and the brain and muscle, I would like to pick up those, and that's what we've been doing. We've been picking up those.
Okay, cool.
In our analysis, yes.
So,
but beyond that, if you say
no,
but your question was that if I pick up, say the medial, suppose I do a medial rectus resection, and there is some amount of medial rectus tissue that comes out of it, which is normal. It's a biological waste, and then I take that and sequence it, right? And then I see. So that's the parallel study that can be done. Yes, no doubt about it, and we can see. But first, we needed to know from blood as to form a general idea of genes. See, we knew nothing. So if you know nothing, you got to start somewhere. That's what, the king said, right? When you don't know where to begin from, there is one place you have to begin from.
Zia, you're an ophthalmologist. You know clinically where the phenotype is. It's
no, that's true. The phenotype is mentioning, let's say, for example, if we talk about strabismus, right?
And obviously, it's in the eye. I'm not telling it's a heart or a kidney or a lung. And you already saw the phenotype.
No, but you see, it's the phenotype. So, okay. Suppose you were talking about
I understand. Glaucoma. I totally
then definitely it's a structural thing. You take up tissue from the angle, and say maybe when you're doing a surgery, when it's a biological waste, kind of a thing. So you can take up that tissue and you can send it for sequencing, no doubt. But in strabismus, we do not know whether it's muscular or whether it's neural. So then where do we start from?
See, this is exactly where I'm going to be in my next question. So now you have sequenced and narrowed it down to 8 genes, and then did you try to find the targets for those genes in the database, the targeting databases?
Exactly.
That will tell you the functionality.
That is what we've been doing. We've been finding out the functional and the gene expression and the targets of those particular genes, and collating it further. So even those 8, if they've come back to 1 or 2, it's because of that. You can eliminate the ones which are not very useful and just take those which actually show a high level of expression. That is the only way that we have gone on till. That's why I said application of logic and filters. That's the only way that we have gone on so far. But having said that, it is not a substitute for actually doing it in tissues or animals. But then I'm not in favor. You can't, for example, strabismus, there's no point talking about animal studies at all, because it's only shown by primates, so you can't replicate in an animal model. But you can of course go ahead with gene expression studies subsequently, but at least, with those two. So what I would propose to do in a subsequent study would probably be, to probably do a mutation screen, basically do it in a large number of population and see whether, it kind of, shows whether, whether the variants, try and find out an association which would be supportive for what you found out on your initial NGS study. So that is the way I believe one would go about it, or one should go about it at this point of time, because it depends on what particular tissue you are talking about. Are you talking about rheumatoid arthritis? Yes, pick up synovial fluid and do it, because that is the actual tissue which is getting affected. So maybe that's better than blood, but here we do not know what is getting affected when we're talking about strabismus. Now, if I'm talking about, say, ARMD, or I'm talking about the retina, then maybe picking up some amount of retinal tissue if it is ethically possible. It may not be ethically possible or whatever at times, because there are many things which are not. If it's a living person, you can't just go about. You can do it in a, person who is no more. If the person has donated his or her body, that is different. But you see, there are many considerations to what you could be doing. So when you're on the other side, you just cannot have a tissue just like that. You can only have waste tissues. And what is actually a waste tissue, for example, like as I said, I resect a muscle, and that muscle is a biological waste, and that can be used, no doubt. But not that you basically use a tissue just to get a tissue for research, and that is not ethically permissible, and it shouldn't be done also. So that's the whole point. True. So blood is basically what, is something which is not. So that is why it's the blood and even saliva for that.
Because there was a paper published saying that, when you do the whole blood and then go to the tissue, they are so much different.
That's true. But how would you get the tissue? First of all, you should know which tissue.
I understand. I totally understand that it's not possible.
You should have accessibility to the tissue. It should be waste tissue.
It has to be a waste tissue.
It has to be a waste tissue. You can't just, kind of. But then yes, if you can
also,
get out of waste tissue, then no doubt. Yeah. Now you tell me which neuronal tissue, which cerebral tissue you would get, or which only resected muscle. And we can try that sometime. Only resected muscle. Yes.
Based on the issues of ethical.
Yeah. So that is the idea is well taken. The point is well taken.
I'll definitely go with the. You said like you have a nice lab setup, and I will definitely go with the stem cells what you're talking about, or some neuronal cells which can be cultured in the cell in the laboratory setting, and introduce those variants inside and check the functionality of those. For example, like muscles, right? So there are a lot of cell lines available.
Exactly.
Different vendors where you can test.
Really be wonderful, actually.
Yeah, so that will help.
That is what we need to do next.
Yeah, that will help you.
That will help
immensely. Functionally, and then also find the targets easily without any human in need of a human tissue in there. So that will help us in two different ways. Like first, you can establish that you can in vitro some model settings, and you can establish in vitro model settings, and as well as extrapolate that to the clinical and the genetical aspects what you're
absolutely
what you already have.
Exactly. The interpretations would be so much more collated. As I said, it's a lifetime of work. As you well know, we talked about that earlier also, and that is exactly how it should proceed.
Much more feasible.
But first is to delineate those 7-8 from blood, and yes, that is true. Yeah, and then work on them. I mean, that's what we plan. But you could of course take in tissues independently, waste tissues related to the particular phenotype you're looking for, and then kind of assess that for seeing what genes come out of those, what areas come out of those. No doubt about it.
Yeah.
It's a good idea for Rolly also, for Rolly and me also. Yes, no doubt. So, yes. Anyway, this was a very initial study wherein we were trying to look at because for a strabismus, like a lot of things are, I mean, most of the things it's not known, this disease.
No, that is the reason why.
Yeah.
So first we wanted to figure out what could be the putative variation would be, what putative genes we could take it forward, and definitely, as you said, that the way forward is to do functional studies. That is very correct. If you want to take it to clinical interventions, then you need to perform those kind of studies.
Yes, and then only can you form a panel, and then you can have a plus minus. So maybe in another 10, 20 years, as I said, it's a lifetime's work, but yes, it's there. All the scope of it is there.
True.
Certain things you take up as a lifetime's work, when you start it off and you kind of go on with it. That is how it is.
It takes time though. It takes time.
It takes a lot of time to come up with genuine results for whatever. Yeah, and the interpretations there.
then the thing is, you publish it, you give people also things to work on. I mean,
Yes,
maybe
exactly.
Zia will do it, somebody else will do it, right?
Exactly. So that is how it is. So when you say about Sanger doing the mitochondrial thing and the bacteriophage, that was the basis of the Human Genome Project. Or if even if Sanger didn't do it, he has actually changed the way you do things nowadays. So that is such a lot of difference to the way things can be done. So it is not that you would be able to do it in your lifetime, but what you start up would probably go on. So that is the whole idea of maintaining the continuum. That is very
True.
important in science.
Yeah.
I mean, it's not that you would be able to do it because none of us are immortal, but yes, the work remains. That is a very
Yeah.
important thing. That's true.
So any more further queries? I think we
No.
had a
Thank
good
you. Thank you very
discussion.
much. Yeah. We'll talk more about.
Yes, we'll talk about this.
Yeah.
Kumar and I
Oh,
had really been interested initially, but there had been other places. But
I
yes,
know. I know. I know. I know. It's
I
not
know.
easy.
We'll
That
keep that.
easy. Yeah.
Yeah,
yeah.
We'll catch up soon.
we'll
Take care.
catch up. Yes,
Thank you.
yes.
Have a
Okay,
nice day.
thank you.
Yeah.
Thank you everyone so
Take
much
care.
for
Bye bye.
attending this talk. It was an interesting discussion, interesting talk.
And if you
And
want then on the specific topics which people want, but those would be more collated and more, more scientific in terms of genetics. I tried to make this more general so that it interests everybody, especially pedigrees and everything, which is, I think, very important to understanding the
yeah,
clinical
yeah,
milieu.
we'll definitely do more to come. So yeah, definitely. So we'll end this meeting now, Zia.
Yes, we've gone over time by about half an hour.
Oh,
But
thank you so much. Thank you. Bye bye.
thank you so much, Raleigh.